So in this video, I'm going to talk about amino acid side chains. So here's the amino acid we were looking at in the last video, right? And so we have one amino acid here, 2, 3, 4, 5. And off of the central carbon, we have these R groups. And we really didn't talk about those R groups in much detail in the last podcast, but they're very important and they participate in protein folding and function. And this R group is what gives diversity to a protein. It's what gives that protein its function, its ability to fold into particular shapes that are important for its function, and so on. So these R groups are very important. And we're going to talk about how to categorize them in ways that tell us a little bit about their function. So 20 amino acids differ only in their R group or side chain. So right, we have an amino group, central carbon, carboxyl group. Only that side chain is different between all the monomers of amino acids. And the properties of amino acids can be determined by their R groups. So side chains can be grouped into electrically charged, either acidic or basic. So the acidic are negatively charged and the basic are positively charged. Uncharged and polar or nonpolar. So these are the four types or three ways to group amino acid or R groups, and we're going to talk about how to recognize those. So here is the chart from your book, figure 3.2, that lists out all of the amino acids with their R groups. And you can see they're grouped into acidic, basic, polar, and nonpolar. And in class, we're going to practice recognizing these, but I also want to point out that on the exam, you'll be expected to figure out which groups they belong to and how they might behave in a protein. So best to learn it now. So the polarity and charge of an R group affects its solubility. So charged and polar side chains are hydrophilic, right? So they interact with water. And those nonpolar side chains are hydrophobic, so they don't want to interact with water. So if you're looking to group the R groups, there's a series of questions you need to ask. And if you ask these questions, you'll be able to really easily group your R groups into specific categories. So I'm not going to be asking you to memorize specific ones, but just to be able to group them. So the first question you might want to ask is, does the side chain have a negative charge? Okay. If so, it has lost a proton, so it must be acidic. So here is aspartate. So we have amino group, central carbon, carboxyl group, and here's the side chain, okay? And you see that this oxygen has given up a proton into solution, so it has a negative charge. And same for the glutamate here. This oxygen has donated, or the hydroxyl group donated a proton to solution, and now it has a negative charge. So these two are your acidic amino acids. And these acidic amino acids, because they have these charges on them, will interact with water because water is a polar molecule. So they're hydrophilic. The next question you're going to want to ask is, does the side chain have a positive charge if it didn't have a negative charge? If it has a positive charge, it's taken a proton on. So it must be basic. So here are a couple examples of amino acids and their R groups that have a positive charge. So lysine, arginine, and histidine all have a positive charge. So they've taken on a proton, so they must be basic. Again, these have charges, so they interact with polar molecules like water. So they're hydrophilic. They're also hydrophilic. The third question, after you ask the first question, the first two, does it have a charge on it? The next one you want to ask is, does it have oxygen? If so, the highly electronegative oxygen will form a polar covalent bond, and thus the side chain is, although it's uncharged, it's still polar. And so you just look through your molecules for oxygen. So serine, threonine, tyrosine, asparagine, and glutamate all have oxygen. So these all create partial charges on the oxygen. And these partial charges create polar side chains. And the polar side chains interact with water, so they're hydrophilic. If the answer to all these questions is no, and then it's a non-polar amino acid. Okay, so glycine, the amino acid we talked about in the last video, alanine, valine, leucine, isoleucine, methionine, cysteine, phenylalanine, tryptophan, proline have no charge in their R group, and they have no oxygen. Okay, so they still have the charge on their amino group, but not within the side chain. So because these don't have a charge and they're not polar, they're hydrophobic. Okay, so they don't like to be around water. So often these non-polar side chains are found, once a protein folds, they're found in the center of the protein's three-dimensional conformation. Cysteine, I want to point out, is also special within the non-polar side chains, and it has some special abilities. So this whole sulfohydryl group is able to form covalent bonds with other sulfohydryl groups in different R chains. So the cysteines create covalent bonds within a protein once it folds. So in summary, you want to ask yourself these questions. Did the amino acid or the R group donate a proton to solution and carry a negative charge? If yes, they were acidic. Did they gain a proton and carry a positive charge? If yes, they were basic. Do they have oxygen? If yes, they are polar. Are all the answers no? If yes, they are non-polar. And bonds are formed because of these groupings. So acidic and basic amino acids can form ionic bonds with each other. Charged and polar amino acids can form hydrogen bonds with each other. And the sulfohydryl groups can make disulfide covalent bonds. So that's just how the R groups, how we group the R groups and how they participate in bonds. And in the next podcast, I'll talk about structure.